Glucose is phosphorylated by using a phosphate molecule from the conversion of 1-ATP to ADP to form glucose 6-phosphate. Hexokinase enzyme is used to catalyze the reaction. Glucose 6-phosphate rearranges to form the isomer fructose 6-phosphate. This reaction is catalyzed by phosphoglucoisomerase. Fructose 6-phosphate is phosphorylated further to produce fructose 1,6-diphosphate by using another molecule of ATP. The enzyme that catalyzes this is phosphofructokinase. Fructose 1,6-diphosphate, a 6-carbon compound, is not stable and it breaks into 2 molecules of 3-carbon compound known as dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. This is catalyzed by isolase. Both hydroxyacetone phosphate and glyceraldehyde 3-phosphate are isomers that can be interchangeable. Dihydroxyacetone phosphate is converted to glyceraldehyde 3-phosphate. This reaction is catalyzed by phosphotriose isomerase. Both glyceraldehyde 3-phosphate molecules are oxidized, hydrogen atoms are removed, and NAD positive is reduced to become an NADH. Phosphoglyceraldehyde dehydrogenase enzyme catalyzes this reaction. An inorganic phosphate is attached to form glycerate 1,3-diphosphate. Both glycerate 1,3-diphosphate lose a phosphate molecule to produce 2 molecules of glycerate 3-phosphate. The reaction is catalyzed by phosphoglycerokinase. Each glycerate 3-phosphate is rearranged to form glycerate 2-phosphate. Phosphoglyceromutase is used to catalyze this reaction. Both glycerate 2-phosphate convert to phosphoenolpyruvate with the loss of 2 water molecules. This reaction is catalyzed by enolase. Both phosphoenolpyruvate lose a phosphate molecule each to produce 2 molecules of 3-carbon compound known as pyruvate. 2 more ATP molecules are synthesized with the phosphate molecules released. The net reaction produces 2 ATP during the glycolysis.
The Krebs cycles occurs in the mitochondrial matrix. The acetyl-CoA combines with the oxaloacetate in a condensation reaction to form citrate. A coenzyme is released. The reaction is catalyzed by citrate synthase. The citrate rearranged by the removal of water molecules and the addition of water to a different carbon atom to form its isomer isocitrate. The reaction is catalyzed by aconitase. Isocitrate is oxidized to form oxalosuccinate. NAD+ is reduced to become NADH. The reaction is catalyzed by isocitrate dehydrogenase. Oxalosuccinate, which is still bound to enzyme isocitrate dehydrogenase, is unstable. It immediately undergoes decarboxylation, loses a carbon dioxide, and is converted to alpha-ketoglutarate. Oxidative decarboxylation of alpha-ketoglutarate takes place. This produces succinyl coenzyme, carbon dioxide, and NADH. A second oxidative decarboxylation takes place. The alpha-ketoglutarate is decarboxylated and the carbon dioxide is removed. The remaining succinyl group join with coenzyme A, forming succinyl coenzyme. NAD is reduced to NADH. The reaction is catalyzed by alpha-ketoglutarate dehydrogenase. Next, substrate-level phosphorylation takes place. Succinyl coenzyme A is converted to succinate. The energy released is used for phosphorylation of GTP, forming GTP. GTP transfers its phosphate group to ADP, forming ATP. The reaction is catalyzed by succinyl coenzyme A synthase. The succinate is oxidized to fumarate. 2 hydrogen atoms are transferred to FAD to form FADH2. The reaction is catalyzed by succinate dehydrogenase. Fumaric becomes hydrated by addition of water is then converted to malic. In this state, the reaction is catalyzed by fumarate. Malic is then oxidized to regenerate oxaloacetate, and NAD+ is reduced to NADH. The reaction is catalyzed by malic dehydrogenase. Oxaloacetate can be used to combine with acetyl-CoA, and the cycle is repeated.
MADH plus H plus is oxidized into MAD plus by transferring 2 hydrogen atoms to MADH dehydrogenase complex. In MADH dehydrogenase complex, FMN will receive the hydrogen atom and is converted into reduced FMN. The reduced FMN is then oxidized when 2 hydrogen atoms are transferred to the electron carrier ubiquinol. 2 hydrogen atoms released from the reduced ubiquinol are split into 2 protons and 2 electrons. The 2 protons produced enter the intermembrane space, while each of the 2 electrons are accepted by Fe3+ in cytochrome reductase complex, which is reduced to Fe2+. Fe2+ is then oxidized by releasing the 2 electrons to another electron carrier, cytochrome C, which also contains Fe3+. When the cytochrome reductase complex passes electron to cytochrome C, another 2 protons are pumped across the inner membrane into the intermembrane space. In cytochrome C, Fe3+ receives the electron and is reduced to Fe2+. The electron from cytochrome C is then released to the third enzyme complex, cytochrome oxidase complex. In cytochrome oxidase complex, CO2+ is reduced to Cu+. Another protons are pumped across the inner membrane from matrix into intermembrane space. The last acceptors of 2 electrons released from the reduced cytochrome oxidase is the oxygen in matrix. Oxygen combined with 2 protons from the matrix and 2 electrons from reduced cytochrome oxidase to form water molecule. The accumulations of H+ in intermembrane space create a proton gradient between the intermembrane space and matrix of mitochondria. The inner membrane of mitochondria is impermeable to H+ ion. As a result, H+ ion re-enters the mitochondria matrix by passing through a specific proton channel called ATP synthetase enzyme complex in the inner membrane of mitochondria. This results in the synthesis of ATP from ADP plus phosphate on the ATP synthase complex using the potential energy released by the movements of H+ ion. This process called chemiosmosis. In the process of oxidative phosphorylation, 1 molecule of MADH will yield 3 ATP, while 1 molecule of FADH2 will yield 3 ADP.